All right. Hello, my name is Mahin Chowdhury, and this is the purification of DNA origami nanoparticles. Firstly, I would like to thank Dr. Veneziano for helping me with this project from start to finish, and I would also like to thank the Oscar program for providing me with this wonderful opportunity. So the overview of the project. DNA origami nanoparticles is a new vaccination method that represents the next generation of vaccine delivery to fight infectious diseases. The current method towards purifying these nanoparticles relies upon ultracentrifugation using AmiconUltra filters, which typically result in a low yield. In a laboratory environment, this is pretty standard, pretty all right. However, if you were to scale this up to the industrial level, it becomes a lot more complicated as this will increase costs to manufacture the nanoparticles itself. So the main objective is to find a method that could help reduce the cost or purify the structures as much as possible. The methods that we will be using. So we will be using 3 different nanoparticles of 3 different sizes, basically, a 6-helix bundle, which is 6HB, pentagonal bipyramidal PB, and pentakelix dodecahedron PD. As I mentioned before, these are different nanoparticles. They have different properties, shaped differently, and have at least 6HB and PB have some similarities in their size. Pentakelix dodecahedron, on the other hand, is a lot bigger. So the best, we will be trying to purify these 3 types of nanoparticles using the methods shown below. The first method we use is the regenerated cellulose membrane filter. As you can see, we have the experimental concentrations retrieved from the trials and the theoretical that was calculated based on the volume retrieved from the end of the trial. As you can see, there is some differences between the theoreticals and the experimentals. I encourage you that you please pause and look through these diagrams. And these diagrams would be compared to the the agarose gel that we collected via gel electrophoresis. These bright bands are the nanoparticles. These bands below are the staple strands or any byproducts that may come off from purifying the actual nanoparticles. These are what we're trying to get rid of, essentially. So as you can see, the brightness indicates the concentration of both the nanoparticles and the staple strands. So the brighter it is, the more concentrated it is, meaning there's more of it, there's a lot in the actual solution. As you can see, the 30K, based on each structure, you can see that each band grows fainter and fainter, for the staple strands at least, grow fainter and fainter as you go up the scale for the filters, which indicates that the filters do purify out as much as they could of all these staple strands and byproducts. Moving on to trial 2, we can see that it is also a little bit consistent compared to trial 1. And if you compare it to the electrophoresis or the gel, you can see that these bands become faint or very barely visible, which shows that it has become very pure and could often indicate that based on the comparison with the theoretical and the experimental, we can determine whether or not we lost the majority of the nanoparticles or we retained them, in which case some we lose a lot and some we aren't able to purify as much. Moving on, the spin polyethylsulfone filters. These filters are used in a similar method with the cellulose filters. As you can see, you have the 6HB, which is similar in theoretical and experimental. However, if you were to compare it to the gel, you have a lot of staple strands. So you can't always rely upon one figurine. You have to kind of you have to kind of compare both. In which case, you can see that many for trial 1 at least, many of these are not that pure. Moving on to trial 2, using the same gel, it's pretty consistent, not as pure as it would seem, especially for PD since the bands are very bright. The spin clean columns by Biorad, they have a very high theoretical concentration, but a very low experimental, which typically indicates that we lost a lot more of the nanoparticles. And as you can see here, we actually have a lot more nanoparticle. We have a band of nanoparticles, but not a band for the staple strand, which means that we were able to purify it. However, at the cost, we lost a lot of the product. The same is consistent with trial 2, in which case trial 2 also had some the same consistency as trial 1. We lost a lot more of the nanoparticles, but we were able to purify it as much as we could. Moving on to this Ziva spin desalting columns, we could see that the experimental concentration is really high compared to the theoretical concentration. And this is also backed up by the bands, the staple strand bands that we see here, especially for PD, which are pretty bright, indicating that these are not as pure as it should be. The same thing goes with the trial 2, same consistency. It just shows that these columns are not a viable method towards purifying the nanoparticles. So the conclusions. Based on the data, many different nanoparticles are best purified by a specific method. PD, for instance, is very well purified using the 100K kDa cellulose membrane filter. There is still some excess that you may have, but you still have a good amount of nanoparticles. The same goes with the other nanoparticles. They all have a different type of purification yield, depending on whether or not the method, whether filter or column, can purify based on its structure and size. So as stated before, some methods would just result in a semi-pure structure, but with a high yield. Other structures have a very pure structure, but a low yield. So is this optimal for industrialization? Not as much. It would be better. It could be better, but mainly because, and this is mainly because we don't know if there are any, we just don't have a very viable method at this point. Right now, the current method is still the best method. But on the other hand, we were able to find that many different structures have there's a better way to purify each structure based on a different method that we can use. So what's next? The next step is to continue searching for a better solution. This is not the end. This is merely the beginning of finding a better way to purify these nanoparticles. Depending on the size or shape, we just have to find something that works for that specific nanoparticle. Thank you so much for listening to my presentation. Please leave any questions if you have any. Once again, thank you so much to Dr. Veneziano and the Oscar program. And thank you again for listening to my presentation. I hope you have a great one.